everyone. I'm Cristabelle. I'm an instructor at Stanford and got a lot of support from DTRF as a early career investigator. And I'm going to talk to you a little bit about the story that we've been working on over the past 2 years. It's very translational and not clinical, so I'm a little like, I don't have patient data, but I want to get your feedback and, have a exciting discussions. I'm a multiomics expert, and also I develop antibodies. So a lot of this talk is focused on multiomic profiling of desmoid patients and the kind of cell populations that we discovered. Also happy to share all our data sets because we've profiled a large number of patients. And So again, I'm preaching to the choir here when we know that desmoid tumors are locally infiltrative, sporadic, hereditary, high recurrence associated with beta-catenin and APC mutation. And when we did RNA sequencing, at the time, bulk RNA sequencing, we found that there were 3 pathways, right, that were enriched in patients, which are the EMT transition pathways, cell migration pathways, and extracellular matrix pathways, which is expected because it's a disease that's, driven by fibroblasts. And in addition to beta-catenin, we found that CGEN, or AP1, was one of the major transcription factors that was driving this fibroblast milieu. And so the question we asked was, when we look at these profibrotic pathways, right, how many of them are driven by AP1? And because we don't want to target GEN because it is a transcription factor, the question was, what are the downstream immune targets that we could discover that we could target? So again, here, this is some of our RNA-seq data that shows the ECM pathways that are upregulated. No surprise there, but it was nice to be validated. And then we found that GEN in the desmoid tumors was driving several other downstream proteins. But because at the time it was RNA-seq, I wanted to ask the question of what are the other target genes that CGEN is driving, and can we develop effective therapies to revert this phenotype? So a year ago, spatial transcriptomics and proteomics became a thing, and we had a lot of, well, not a lot of, but we had patient samples, and so we built a TMA. And the question I wanted to ask was, in addition to fibroblasts, what are the other cell types that we can see when we look at patients, right? So we performed spatial transcriptomics, both at the transcript level as well as the protein level. At the transcript level, it's about 1,000 targets, and at the protein level, it's a little, smaller. So this was the TMA that we built. And what we found was that these desmoid tumors, we've always been just focusing on the fibroblasts. And what we found was that there are these immune subsets and these ECM cell types that are very distinct cell populations that we could see in these patients. And so if you look at these distinct cell clusters, right, in the patients, you see there are 5 different cell types. And we compared these between desmoid tumors as well as normal tissue. And interestingly, we found that there was this very, heterogeneous fibroblast cluster where the cell types were not exactly fibroblasts and not really epithelial, and they had this ECM-like phenotype that we wanted to, look into. And we found that they were both expressing PDGFR as well as GEN. And if you look at these clusters, you can see that in the patient samples, you can see a high amount of GEN as well as PDGFR. The other distinct population that we found was these macrophage populations, which again, nobody has looked into, especially with regard to the innate immune component in patients. And we found that, patients had a high number of M2 macrophages, and relative to normal tumors, normal donors that had M1 macrophages. So now we knew that in addition to fibroblasts, we are also looking at an innate immune component and ECM remodeling for patients. So then we wanted to deconvolute this macrophage subtype, and try to understand what are the different types of macrophages we see in patients' samples. And we found that there were 6 different types of macrophages. And this is a novel subtype, and we never seen this before. And the question that we wanted to know was what is a potential immune molecule that is driving these macrophage populations. So again, here you see between the normal and desmoid tumors, you have a distinct expression of M2 macrophages. And this was a surprise to us because we didn't go in thinking that we would find, unique macrophage populations. So we call them disease-associated macrophages, mainly for the reviewers. And we found that one of our targets of interest, called CD63, was elevated in these macrophage populations. And these were distinctly enriched significantly between patients and the donors. And then we did single-cell RNA sequencing in the patient samples and combined it to look at CD63. And we found that when we focused on the patient populations, that 63 was enriched in the macrophages as well as some of these fibroblast populations. So what is CD63? It is a tetraspanin that is regulated by c-Jun, and that we know because we've done a lot of work that I'll show you. And one of my goals was to develop an immune therapy that could target this protein. There are several other targets also that we picked up in our multiomic profiling that I'm happy to share with you if you want to, look deeper into this. And so then we got 2 different cell lines from Mushrik and Jesse, and I think it's originally from the Pollock group, that we performed flow cytometry on to check for the expression of our marker. And then I wanted to see if we overexpress Jun and overexpress 63, does this lead to an increased growth dynamic in the desmoid patient cells, in the primary cells. And indeed, we see that there is a significant increase in proliferation over time with the overexpression of both c-Jun and CD63. And then I wanted to see if Jun is indeed transcriptionally regulating 63, and I did some genetic knockouts, and you can see that when Jun is knocked out, we have a decreased expression of CD63, and when it's overexpressed, you see an increase, and you don't see this effect in TGF-beta, which is, a traditional marker of fibrosis. And so the next question I wanted to ask was, is this true? And so we performed single-cell ATAC-seq in the primary fibroblasts of the patient samples that we had in the primary desmoid patient samples. And so when we knocked out Jun and beta-catenin, we saw a decreased expression of decreased binding at the promoter of 63. And we also saw that, for beta-catenin as well as 63, there was a decreased binding at the promoter region. So we know that Jun is potentially collaborating with beta-catenin at the CD63 promoter and driving its expression. And then I confirmed these results using chromatin immunoprecipitation, where when you knock out Jun, you see that there is a decreased binding at the 63 promoter. That's confirming that AP1 is indeed driving this immune protein, which in turn is driving this ECM matrix that could, that is promoting this profibrotic milieu in desmoid tumors. And then when I knocked out Jun, I saw that there was a decrease in other profibrotic markers like FSP1, collagen A1. And when you knock out Jun and 63, it reverses this profibrotic gene signature that is seen in primary desmoid cells. So we know that by knocking out 63, we can reduce this profibrotic phenotype, and when we overexpress it, we get this profibrotic phenotype. So mechanistically, we know that AP1 is transcriptionally regulating 63, which is indeed driving this profibrotic signature or this profibrotic pathway that causes, your desmoid cells to grow and, proliferate. So the question was, we have the mechanism, we have a target, we have omics data, but what does that mean? So we developed a monoclonal antibody that we're now trying to convert into a recycling antibody. And the question was, can we target 63 for a potential ECM remodeling to be used as a combination therapy for DTF patients. And here is some data from the antibody that was generated. We compared it to some of the commercial non-humanized antibodies for expression to show that it does, bind and block to CD63, block 63. And what we showed that was when we use our antibody, it blocks 63 and induces phagocytosis. So it inhibits this crosstalk between a diseased fibroblast and a diseased macrophage and induces phagocytosis. So you're eliciting an innate immune response to cause, your pathogenic fibroblasts to be eaten, right? And so we found that Seagen does transcriptionally regulate 63, and we can block 63 as a potential strategy to remodel the extracellular matrix to treat DTF by activating the innate immune component. And so, yeah, we have, in summary, we have a lot of multi-omic data that identified 63. And there are other immune molecules that, if you guys are interested, please, I'm happy to share this data. We've identified very distinct cell clusters, in particular, these ECM cell clusters and 5 different macrophage, disease-associated macrophages that we can look into. We know that Seagen is an essential promoter of this pathogenesis, and overexpression of Seagen in primary desmoid cells promotes this diseased phenotype, both in vivo, ex vivo, in vitro. And then we have an antibody blocking therapy to block CD63 and elicit an innate immune cell function. So with that, I would clearly have used my acknowledgement slide from last year, but this is our group at Stanford. I'm now in a more independent position, happy to take any questions, discussions, collaborations, and looking forward to share data and look into, have a more multi-omic approach to target identification and distinct cell type classification in desmoid tumors. Yes.
Very, very cool, very interesting.
Sorry, it's very scientific. I feel like this all.
No, I like science. But so in your cartoon, it was in CD63 was interacting with the receptor on the macrophage that you tried to block. So what would be the receptor?
So galectin. We're thinking that galectin-1 is one of its binding partners as well as TIMP1. But right now, what we're trying to do is 63 is present on the disease-associated macrophages and also on these fibroblast populations. So we're trying to elicit this, macrophage reconfiguration to switch from like the M2 to M1 phenotype. But we're still trying to figure out what's the best way to do it because we want to target both the macrophages, the inflammatory pathway in the macrophages as well as the diseased fibroblasts. So the first way to do it is through phagocytosis, but we're also developing an alternate, therapeutic approach to switch this macrophage phenotype. Yeah. Yes.
Are you thinking of trying to modulate macrophages by CSF1R inhibitor drugs, or just a way to?
No, I mean, we are thinking of it, but I'm an antibody development person, right? So I did want to bring something novel out there, but we are comparing it to, CSF1.
Thank you.
Yes. Yeah, we're collecting the animal, the tumors and the animal data to do omics, but right now all the omics we have is in patients, and so, But that's the next step. We have to do it.
So what is the source of the CD63? Is it from the macrophages, from the stroma? Where does it come from? It's
from the stroma. So I think it's from so right now we see a high expression in the macrophages, but we also see expression, in the pathogenic fibroblasts. So when you look at the tumor, the desmoid tumors, you see, you have the normal fibroblasts, and then you have, the pathogenic fibroblasts. And 63 is one of those, immune molecules that is, seen in the pathogenic fibroblasts, and it's associated with, disease progression. So if you look at the trajectory mapping, 63 and Jun are associated with like disease formation. We don't know exactly, it's a tetraspanin, so it's, extracellular. We don't know what is the cell type that releases it, but we know that, it causes a switch in the macrophages, and it's also associated with the pathogenicity of the fibroblasts. So I'm thinking that this blocking 63 would be a good way to remodel the ECM, which can be used in combination with like gamma secretase inhibitors, right? Or small molecules where, the ECM is a problem for desmoid tumors. Oh, yeah.
So the blocking of the macrophage may be another approach, right? So rather than blocking 63, block the effects of 63 in the macrophages.
Yeah, I think.
Are desmoids infiltrated heavily with macrophages? I mean, they're so inert, right? Do we know that? Is there a big macrophage infiltration that would be a druggable target in desmoids? Do we have a pathologist here? I need a pathologist. Who's a pathologist?
We didn't know that. This was a surprise.
Because, I mean, that would be important, right? Because if it's really driving macrophages, then you'd have to have a macrophage population to target. If C63 is driving the macrophages, you'd think there'd be an infiltration of macrophages. It's an interesting thought. I'm just want to understand.
Yeah, I think like we've not been looking at additional cell populations. I think looking at desmoid tumors, we're just looking at fibroblasts, right? And it's not just that. It's like, it's the ECM, and how does this communication proceed? We've not looked at macrophages, right? And so when we did the spatial omics data, I actually was very surprised. I'm like, wow, what are these cell populations? And that's why I would love if you guys want to play around with the raw data and, combine it with what you have and, see this thing, because it's new, it's, something that we've not looked at before, and we've not thought of combining ECM remodeling therapies, yeah.
Okay.
What other cell types express CD63?
It's expressed at a very low level in some epithelial cells, but it's also an exosome. It's an exosome marker. Traditionally, it was used as an exosome marker, but we've seen that it's very significantly enriched in the pathogenic fibroblasts as well as now in these macrophage populations. So that's especially in desmoid tumors. So originally, it was discovered in HCC, which is another project that I was working on. And, I just wanted we ran the omics profile, and I said, well, let's look at these targets, and it showed up in desmoid tumors, and I was like, wow, okay. So, yeah, but it is expressed at a low level in several cell types, yeah.
So in the case of the fibroblast, do you know if it's expressed in the membrane or is exosome expression?
It's on the membrane, yeah,
yeah.
We did a lot of, to look for extracellular expression. We did a lot of FACS to look at it, localization.